With this video, we're going to try to make a little bit of sense about what happens when light strikes an object. So some of the things that occur as we work to try to explain what's going on inside a microscope and how to mitigate some of the issues related to light microscopy. So here we go. So when light strikes an object, one thing that can happen is what we think of as reflection. So imagine going and looking at the mirror and seeing your lovely face in the mirror. That is reflection that you're observing. The light energy bounces off the surface of the object, in this case the mirror, and it's actually double reflection if you're looking at a mirror, because the light is bouncing off your face, going to the mirror and bouncing off your mirror and coming to your eyes. But if we check out this image here, the top one, I love this. I love this image. It's awesome. So here's a light source. We've got a candle burning. The light energy strikes a smooth surface and then bounces off of it. And some of that light energy bounces into the dude's eye. And so he is able to perceive it then. And so when we see green plants or a red t-shirt, what we're seeing is the green wavelengths bouncing off of or being reflected from that green plant, or the red wavelengths of light being reflected from that red t-shirt. And all the other available or perceivable wavelengths of light, they have been absorbed by the red t-shirt or by the green plant. So we'll talk about absorption in a minute. So materials reflect light. They can also transmit light. With light energy being transmitted, what we're talking about is, say, like a pane of glass. So if you look out a window wherever you are, what you're seeing is the light that has been transmitted through the glass and to your eye. So here comes the light energy, and it passes unchanged through the light, and that is transmission. So panes of glass transmit light. The microscope lenses also transmit light. And so as light passes through the lens, it's being transmitted. Okay, so reflection and transmission are two things that can happen when light strikes an object. The third thing that can happen is absorption. So this is when the light energy is taken up by the pigment molecules in the object. So plants, for instance, absorb the non-green wavelengths of light, and they use the energy in that absorbed light to photosynthesize, to convert that energy into glucose. And some of the energy is also then converted into heat in that process as well. We'll talk about energy conversion in just a second. But absorption then is the idea that the light can come and it gets, these little squiggly lines here are showing the energy being absorbed by the molecules in whatever material it is that the object is made out of. Now there's some cool things that can happen when light gets absorbed. As some molecules, not every molecule does this when it absorbs that light energy, but some molecules are capable of storing the light energy and then re-emitting it. And so the explanation for how this kind of luminescence occurs is a little bit complex. But here we see the light energy coming into a molecule, in this case to an atom in the molecule. And what happens is that an electron, they're showing it here as this yellow electron, gets bumped up to a higher energy state. And so its energy then, that electron absorbs this energy and is still circling around the same nucleus. So we haven't changed the molecule or changed the atom that it has absorbed this energy, but it has become a little bit more energetic because the electron is now circling the nucleus at a higher energy level. Later on, that electron may re-emit all that energy that it got and fall down to a lower energy level. And when it does, it sends out the energy in the form of either fluorescence or phosphorescence. So fluorescence and phosphorescence. And so with fluorescence, as the light is hitting it, the electron gets bumped up to a higher energy level and then immediately falls back down to a lower energy level. And as it falls down, the energy that it is releasing is emitted in the form of light energy, visible light energy again. And so that's how fluorescent light bulbs work, is that we are exciting electrons that are found in the gas inside of the, and at the surface of the light bulb, the fluorescent light bulb, and it is then dropping those electrons back down to their original energy level and re-emitting the light through fluorescence immediately. Phosphorescence is when the molecule can actually hang on to that light energy for a period of time and then re-emit the light later on. So when we think about glow-in-the-dark things, stuff that you put under a bright light, and then later on you can turn out the light and it re-emits re-emits the light energy, that is phosphorescence, because it's happening after the light energy has, the original light energy is gone. So fluorescence is immediate release of that light energy, or that energy again as light. With phosphorescence, it's later on re-emittance of that light energy. So what we see frequently is that molecules can even absorb UV light and then later on re-emit it as visible light. And so this picture that I'm showing here is, and if you've ever been to a rock museum, frequently they'll have an exhibit set up where they're shining UV light on the rocks, and then the rocks are phosphorescing and they're glowing back in these amazing colors. I couldn't find any good images of a before, like invisible light, and then under UV light, the phosphorescence. But you can see how many of the minerals in that sample, or in the picture there, are glowing back a green or a yellow or a red or even kind of a neon-y looking blue color. So luminescence is the absorption of light energy and then the re-emittance of that light energy, frequently in longer, or always in longer wavelengths. So we take a shorter wavelength light and re-emit it, take that in, and then re-emit it as a longer wavelength of light. Okay, so absorption can happen as well as luminescence. Refraction is also a characteristic of many materials. Refraction is when we bend the energy as it passes from one material or one medium to another. And it occurs because of the different densities of the media and their index of refraction. So different density, or materials with different density, tend to have different indexes or indices of refraction. So in the microscope, what we're seeing is that one medium that light is passing through is air. And then when it hits the lens, it gets refracted or bent because the lens, the glass lens, has a different refractive index. And what happens every time that the light refracts, every time that it bends like that, we lose light energy, and the result is that we get blurry images. So one of the ways that we get around this is by using a material called immersion oil. So if we're talking about now the objective lens of the microscope and our slide, so glass now, what we do when we use immersion oil is we put this bubble of oil here on top of the slide in the cover slip, and the immersion oil has the same refractive index of light as light does, and I didn't show it, but it comes into contact with both the objective lens and the glass of the cover slip. So now we have glass, immersion oil, glass again, and there is no refraction at that point. And so because of this, when we use immersion oil. We prevent refraction, and the light doesn't bend, that light energy isn't lost, and the image remains sharper. So doing something to overcome refraction can help produce sharper, better quality images. So here we're seeing refraction occurring. This is a block of plastic. Here comes a laser light. Boom, light is coming, and dun, it refracts, and then it refracts again just a little bit as it comes out of the block. So we see two examples of refraction when we are passing light through this block of plastic. Here we can see that as light is passing through first glass, then through water, and then reflecting off of this glass rod that is suspended from the, and that's what this is. We see this glass rod that's suspended from the lid inside. It even looks magnified now because as we refract things through curved surfaces, it changes the magnification. That's the point of a lens here, is that we're focusing and changing magnification of the light as it passes through the lens. And so that curved glass acts like a lens, and so does the water as it refracts. And so we see a magnified rod when we look down into the water. But if we use immersion oil with no refractive index and no bending of light, there is no, the glass rod becomes invisible. So that is not a retouched photo where they've made the glass rod go away. It's actually, because there is no refraction around the edges of that glass rod, it becomes virtually then invisible, and we can't see the glass rod. And so this is another way, or one of the points, I think the book makes this point, that you can smuggle diamonds in oils like this because it just looks like you have a little container of some oil. And the diamonds, because they have a similar refractive index to whatever the smuggling oil is, they become invisible there in the bottom of the glass jar. Okay, so we use immersion oil as a means of preventing refraction and maintaining image clarity. We also stain subjects, so we'll play around some with staining when we are in the lab this week. And so as we stain, we are changing the index of refraction, which makes it easier to see the edges between the material that's absorbed the stain and the material that isn't stained in the slide. And so refraction can be of use because it helps us to show up those edges in the same way that here refraction has helped us to see the edges of the glass rod. But there is a cost to that refraction, and that is a little bit of blurriness ensues. So working at the kinds of magnifications that we're working with, up to about 1000x, the trade-off is still good enough that the resolution makes it, that we can achieve, makes it possible to overcome this energy loss because of refraction. Okay, so refraction can occur. Diffraction is another thing that can occur as light passes around the edge of a material. So like if you're looking at something here that has an edge, as the light bends around the edge of it, it gets broken up into the rainbow that we see. Or as it passes through a hole, it creates this, it bends and distorts the light. So it can cause this rainbow halo effect around an object. Diffraction can. And it can cause blurry edges at the object as material, as light is passing through small apertures or small holes like this. And the lenses act like small holes. And so the more that we magnify an image, the smaller the hole we're looking through effectively, because the lenses are bending the light into ever tighter and tighter points here. The more that we magnify, the more the light is bent into a tighter little point. and that causes refraction and diffraction and blurry images. So that's why the greatest useful magnification that we can achieve is about 1000x, because all of this diffraction that's occurring as we're focusing the light into a smaller and smaller little pinpoint, then the greater the diffraction and the more blurry the image becomes. And so that's when we've got to do, as we're working at 1000x, we've got to do everything we can to up the clarity and the resolution of the image. And that's why we turn to doing the immersion oil, so that we at least aren't getting any refraction causing lots of blurriness. Okay, so we'll wrap up this section then by talking a little bit about the light microscopes and the anatomy and physiology of light microscopes. So we're going to be using a compound microscope. That means that there are multiple lenses within both the objective lens and then here at the oculars. So we're using multiple lenses to focus the light and correct for the distortions that occur in the image as we enlarge it, as we magnify that image. We have binocular microscopes that we're using. That is, that they have two eyepieces or two oculars rather than monocular. There are monocular microscopes too, but ours that we're using, the good ones that we have, are binocular. And they are also parfocal. So what parfocal means is that as you are focusing at a low magnification on this point of the, in this region of the slide, as you change the objective lenses and successively look at smaller and smaller regions of the slide, they are mostly centered. Those ever smaller regions are centered on each other. So you're just moving in like to the smaller portions of a target as you with a parfocal microscope. As well, the objects near the center tend to be close to the same focus. So a parfocal microscope is a good one to use because you remain in about the same place. Whatever you put at the center at a low magnification will also be at the center at higher magnifications. Okay, so some of the anatomy and physiology then of the microscope. The base, it's the support area. The arm is where you carry it. So every time that you pick up and move the microscopes, be sure to hold them at the arm and have a hand underneath the base. The illuminator is the light source here. So there's a lamp in here, and there are mirrors that send the lamp up through this first lens. So that's the illuminator. It reflects light up to the specimen. The condenser is found here. That focuses the light. It converges the light onto the specimen. And so as you move the condenser up and down, so it's got a dial that moves it up and down in this direction, it will focus the light more tightly on the specimen. Inside the condenser, there's also an iris diaphragm, and so that will either enlarge or tighten down so that you can control the amount of light that is passing through the specimen. And it turns out that as you get to higher and higher magnifications, opening the iris diaphragm will bring more light in, so that can be useful. And playing around with that, finding the perfect amount of light to go through, will help you to be able to see the edges of structures. So messing with and adjusting the iris diaphragm is going to be a really useful tool for your microscopy work. There is a mechanical stage. This is a stage that moves around underneath the lenses, and it has on it stage clips. These are the stage clips here. So we've got these stage clips that can also move the slide around. So the mechanical stage is super useful for finding what you would like to look at underneath the microscope. On the nose piece, this is this area, we have 4 lenses. At the lowest magnification is the scanning lens. It provides 4 times magnification. So scanning lens, that's the red one. The low power lens is next. It has 10 times magnification. The high power lens provides 40 times magnification. And then the longest lens is the oil immersion lens. That's where you have to put the drop on, and it gives 100 times magnification. So the body tube is this portion here, and it reflects. It's got mirrors inside and lenses that reflect the light up to the oculars. So the light is coming from here, it gets focused by the condenser, it's adjusted by the iris diaphragm, it passes through the subject, up through the objective where it's focused even more, and the light is spread apart to produce magnification. And then it's reflected through the body tube and up into the ocular lenses. So here are the oculars, also known as the eyepieces. The eyepieces on our microscopes provide 10x magnification, and they do adjust back and forth. So you want to adjust them to match your own interpupillary distance. And so as you're moving back and forth with your partner in front of the microscope, you may have to adjust those two from side to side. They also screw out, and so they can do a little bit more focusing. So if you were just using your own microscope, you could actually adjust them to match the amount of magnification that you require at your own eyes, the correction that you need based on your eyeglass prescription. So everybody's eyes magnify and focus just a little bit different, and you could actually adjust those for yourself. I recommend that you just screw them all the way down and not try to mess around with them between you and your partner, and because otherwise it's just too tricky. And then the focusing knobs here, we have the coarse focus, that's the large one. And recall, you only want to use the coarse focus on the shortest red lens, the scanning lens, is the only time you would use the coarse adjustment knob to provide focus. But that will move you very quickly, move the stage up and down very quickly so that you can get into focus. So you do that with the scanning lens. And then after you move to the low power lens, you would only use the fine adjustment knob from there to the higher magnifications. So that will help you. It only moves the stage up and down just a little bit, giving you fine control over the actual focus. Okay, so there you are, anatomy and physiology of the microscope. We'll be checking in on that little in-class exercise to make sure that everybody's got that got their anatomy and physiology of the microscope down. And then here's our last slide, just so that you have thought about total magnification, because we don't just talk about the magnification provided at the objective lens, but we also have to include in total magnification the amount of magnification that's coming from the oculars. And it's a multiplication function. So 40x is the scanning lens, low power, total magnification of 100x, 400x for the high power, and then 1000x for the oil immersion lens. And they are also coded by color. Red for the scanning lens, yellow for the low power, blue for the high power, and white for the oil immersion lens. Okay, so there you have it. Everything you ever wanted to know about how light interacts with objects and the pieces and parts of a microscope and what they do. You'll want to be ready for being able to explain what the pieces and parts do on the exam, so just be prepared for that. Okay, again, as always, you know how to reach me. Reach out with an email or a phone call to my office, or see me during office hours.